Bacterial spores. Bacterial spores or endospores are metabolically inactive, highly resistant structures produced by some bacteria as a defensive strategy against unfavorable environmental conditions. The bacteria can remain in this suspended state until conditions become favorable, and they can germinate and return to their vegetative state. The spore is surrounded by a thin delicate covering called exosporium. Spore coat lies beneath the exosporium. It is composed of several protein layers and may be thick. It is responsible for spores' resistance to chemicals. The coat is also thought to contain enzymes that are involved in germination. The cortex may occupy as much as half the spore volume. It rests beneath the spore coat. It is made up of peptidoglycan that is less cross-linked than that in vegetative cells. Spore cell wall or coat wall is inside the cortex and surrounds the protoplast or spore core. Core has normal cell structures such as ribosomes and a nucleoid, but is metabolically inactive. Calcium dipicolinic acid is present in core. Process of sporulation. Sporulation commences when the bacterial growth ceases off due to unfavorable conditions. It can be divided into 7 stages. Stage 1. An axial filament of nuclear material forms. Stage 2. Inward folding of cell membrane to enclose part of DNA and production of four-spore septum occurs. Stage 3. Membrane continues to grow and engulfs the immature endospore in the second membrane. Stage 4. Cortex is laid down in space between the two membranes as calcium dipicolinic acid is accumulated. Stage 5. Protein coats are formed around the cortex, leading to formation of spore coat. Stage 6. Maturation of endospore occurs. Stage 7. Lytic enzymes destroy the sporangium, releasing the spores. Significance of endospore structure. The layer-by-layer coating protects the spore's genetic material and other valuable biomolecules from harmful chemicals or unfavorable environment. Spore coat and dehydration of protoplast appears to be very important in heat resistance. Also, the spore coat structure ensures impermeability of unwanted molecules. The cortex protects the core, which consists of genetic material. Calcium dipicolinic acid is resistant to wet heat, oxidizing agents, and sometimes dry heat. In addition, high content of calcium dipicolinate present in the core stabilizes the spore's genetic material. Small acid-soluble DNA-binding proteins found in the endospore saturate spore DNA and protects it from heat, chemicals, and radiations. Germination. It is the breaking of spore's dormant state. This process is characterized by spore swelling, rupture or absorption of spore coat, loss of resistance to heat and other stresses, loss of refractility, release of spore components, and increase in metabolic activity leading to cell divisions. The position, form, and deformation of the spore. Spore can have central, subterminal, and terminal position, can be of spherical or elliptical form. It can be non-bulging or bulging. Importance of endospore. These structures are extraordinarily resistant to environmental stress such as heat, UV rays, gamma radiations, chemical disinfectants, etc. Endospores are of great practical importance in food, industrial, and medical microbiology, as it is essential indicator in sterilizing solutions and solid objects. In the environment, endospores aid in survival when moisture and nutrients are scarce. Spores' position and form may also be diagnostic use by employing Sheffer-Fulton's endospore staining method. The main purpose of endospore staining is to differentiate bacterial spores from other vegetative cells and to differentiate spore formers from non-spore formers. Visualization of bacterial endospores. The Schaefer-Fulton method uses heat to push the primary dye, that is malachite green, into the endospore. Washing with water decolorizes the cell, but the endospore retains the green color. The cell is then counterstained pink with safranin. Endospores appear light green. Vegetative cell appears brownish red to pink. Examples of various positive endospore staining are Clostridium perfringens, Clostridium botulinum, Clostridium tetani, Bacillus anthracis, Bacillus cereus, etc. Thank you for watching the video. Don't forget to subscribe and hit the notification bell to remain updated with more such videos.